Hello everyone, myself Khalid Feroz. My assignment is on Guava breeding. Before starting my presentation, I would like to thank Dr. Prashant Bakshi who started this online program for the students during this period of pandemic and provided us all the needy help regarding our coursework. In this presentation, I would like to discuss the principles and the practices of breeding in Guava. These are the contents of my topic. Introduction. Guava is one of the important fruit of tropical region of world and is known as apple of tropics. Fruit contains vitamin and vitamin B and is highly rich in vitamin C. Guava is hardest among tropical fruits, can stand waterlogged conditions to a greater extent. India ranks first in guava production in the world. Major producing states are Madhya Pradesh, Maharashtra, West Bengal, Gujarat and Karnataka. Classification. Important species. First one is Psidium guava, commonly grown guava, small tree with greenish brown scaly, box slender trunk. Ovary is inferior. Edible portion of fruit is thalamus and pericarp. The fruit, a berry, is a round, ovoid or pyriform, commonly yellow in color, with flesh varying white to deep pink or lemon in color. Psidium cattleianum, Chinese or strawberry guava. It's native of Brazil, ornamental in appearance, and is a favorite garden shrub in many regions. Tree is small in stature, look like shrubs with smooth grey-brown bark. Fruits are ovate to round, purplish, radiate color with a thin skin. Flavor is sweet and aromatic, suggesting that of strawberry. Another one is Psidium fradioricus. It's also known as Costa Rican guava. The tree is tall, having height ranging from 7 to 10 meters. The branches are slender and smooth. Small size fruits with globular shape. Fruits have high acid content, wilt resistant species, and is also used as wilt resistant rootstock. Another one is Psidium montanum, also known as mountain guava. The plants are just like shrubs, attain 1.5 meter height with flat round branches. Fruits are green, subglobulous, about 2 centimeter in diameter and are very fragrant. It's mainly found on mountains in Jamaica. Other one is Psidium pyriform, also known as pear-shaped guava. The tree is small, hardy in nature. It prefers warm climate for flowering and fruiting. Fruit has white flesh. The plants can be easily grown in container for the house or greenhouse. Origin and distribution. It's native to tropical America, stretching from Mexico to Peru. It was introduced in India by the Portuguese during the 17th century. Guava belongs to family Myrtaceae, which has more than 80 genera and 3,000 species distributed throughout the tropics and subtropics. CYTOGENETICS Genus Psidium contains about 150 species all over the world, having basic chromosome number 11. All cultivated varieties of guava in India belongs to Psidium guajava, or either a diploid or triploid. A natural triploid was reported, but most of them are shy barriers. Now, important cultivars of guava mainly grown in India: Allahabad Safeda, L49, Sardar, Lalit, Lalima, and seedless, Shweta, Punjab Kiran, and Hisar Surkha. These are the newly released hybrids of guava: purple guava, Thai guava, and AC 105. Varieties description. First one is Allahabad Safeda. The fruits are big in size, round, smooth skin, white flesh, soft, firm, light yellow, and on ripening develops very sweet taste with pleasant flavor. The TSS of fruit is 10 to 12%. It bears heavily. The yield may vary from 120 to 140 kg per tree. Sardar L49. This is seedling selection from Allahabad Safeda. It is a semi-dwarf tree with 2 to 3.5 meter height, profuse branching type with dense foliage. The average fruit weight is about 175 gram. The seed number is about 340. The second color is primrose with red hot spots on the second, sweet taste and has good keeping quality. The yield may vary from 125 to 150 kg per tree. Lalit. It's recently released variety. The fruits are medium sized with attractive saffron yellow color and red blush. The flesh is firm and pink with good blend of sugar and acid. It gives 24% higher yield than popular variety Allahabad Safeda. Shweta. Its selection from apple color recently developed by CISH Lucknow. The fruits are medium sized 195 gram in weight and globose in shape. The fruits are attractive with creamy white red spots on skin. The vitamin C content of fruit is 300 mg per 100 gram of pulp. The TSS ranges from 10.5 to 11%. It is prolific barrier. Apple color. The fruit is famous due to their color. The fruit is dotted pink color, spherical in shape and sweet in taste with good keeping quality. This cultivar is valuable for breeding program as a donor parent. Seeds are about 278 in number and bearing is heavy. Genetic resource. The greatest diversity of species is from central and southeastern Brazil, but there are also good number from northern South America. 60 accessions were added at IHR, one from exotic collection and one indigenous collection were added at Fruit Gene Bank. In India, many Guava clones have been introduced from different countries like Brazil, Australia and USA, and different species were introduced like Ceratorium, Littoral or Longipicus. They can be utilized in varietal improvement programs. There are about 160 cultivars available in India of guava. Blossom biology. Guava bears flower solitary or in cyme of 2 to 3 flowers on the current season growth in the axil of leaves. Blooming period varies from 25 to 45 days depending on cultivar. About 1 month is required from flower bud differentiation to complete development up to calyx cracking stage. Peak of anthesis is between 5 to 6:30 am. In the most varieties of guava, the dehiscence of anther starts from 15 to 30 minutes after anthesis. There are 3 flowering seasons in guava. One is Ambe Bahar, Bahar and Hasta Bahar. Initial fruit set of guava is very quite high. Formation of fruit is first noticed after 12 days from flowering. It's observed that the fruit originating from the central floral button almost always presents faster development. Breeding system. Guava is self-pollinated crop, bit cross-pollinated by insects at some extent. The pollen fertility is high in almost all the cultivars. The pollen fertility is 78 and 91% in Allahabad round and Lucknow Safed respectively. The maximum fruit set occurs when the stigmas are pollinated within 2 hours after anthesis. For making crosses in hybridization program, flowers are emasculated at least 1 hour before anthesis and bagged. Breeding objectives. To develop high-yielding varieties, precocious dwarf varieties with fruits of uniform shape, good size, attractive skin, pink pulp color, fever and soft seeded, resistant to wilt, long storage life and suitable for table and processing purposes, good branch angles and spreading tree habit. Varieties suitable for cultivation under arid, saline, water, logged marshy, cold areas. To evolve wilt resistant. Genetics of important traits and their inheritance pattern. Commercially important traits such as yield, fruit size, certain types of disease resistance, and quality characteristics are often in the low heritability category. None of these characters are determined solely by major genes, although basic genes subject to the modifying effects of polygenes have been identified for some characters like skin color and acidity. Red pulp color is dominant over white, and this character is garnered monogenically. Many cultivated red-fleshed varieties were found to be heterozygous for this character. Bold seeds in guava were found to be dominant over soft seeds and are also governed monogenically. A linkage was also found between the red flesh color and bold seed size, albeit pear shape of the fruit is dominant over round shape. Triploidy and some other genetic factors were also responsible for the female sterility. Problems in breeding, low fruit set and fruit drop, long juvenile phase, crossing compatibility between several cultivars, limit the combining of superior traits in the single genotype, for example L49, apple color, and Behat Coconut are not intercrossable. Seedlessness is due to triploidy, ovule sterility, or chromosome aberration. Floral structure, epigynous flower with abundant incurred stamens of varied size and long juvenile period. Breeding methods and achievements. First one is introduction, selection, clonal selection, hybridization, polyploidy breeding. And in this, I will discuss clonal selection, hybridization, and polyploidy breeding. Clonal selection. Improvement work in guava started for the first time in the country in 1907 at Ganesh Fruit Research Station, Pune, with the collection of seeds of varieties grown in different places to isolate the superior strains. The first chance seedling strain from the open pollinated seedling of Allahabad Safeda was released with the name of L49. At Horticultural Research Station, Sarhanpur, evaluation of seedling types resulted in a superior selection S1 having good fruit shape, few seeds, sweet taste, and high yielding. At IIHR Bangalore, from 200 open pollinated seedlings of variety Allahabad Safeda collected from Uttar Pradesh, one seedling selection 8, was found to be promising. Hybridization. At IIHR Bangalore, as a result of hybridization among Allahabad Safeda, red flesh, chitadar, apple color, L49, and bananas, 600 F1 hybrids were raised. One hybrid, Archa amulia, has been released recently. It is a progeny from the cross of Allahabad Safeda and triploid. Hybrid 16-1 is a cross of apple color and Allahabad Safeda has been developed recently. At Fruit Research Station, Sangareddy in Telangana, intervarietal hybridization resulted in the isolation of two superior hybrids. First one is Safed Jam. This is a hybrid between Allahabad Safeda and Koir, a local collection from Hyderabad, Karnataka region. It is similar to Allahabad Safeda in growth, habit, and fruit quality. The fruit are bigger in size with good quality and few soft seeds. Koir Safeda. It's a hybrid between Koir and Allahabad Safeda. The tree is vigorous, fruits are larger with few soft seeds and white flesh. CISH Lucknow isolated two hybrids H136 for red pulp and soft seeder with high TSS. Haryana Agricultural University, Hisar has released two hybrid varieties. First one is Hisar Safeda. It's a cross between Allahabad Safeda and seedless, which has upright growth with compact crown. The fruits are round, weighing about 92 gram each. Pulp is creamy, white with less seeds. TSS is 13.4% and ascorbic acid is 185 mg per 100 gram. Hisar Surkha. It's a cross between apple color and Banarasi Surkha. Tree is medium in height with broad compact crown. Fruit is round, weighing 86 gram each. Pulp is pink, having 13.6% TSS, 0.48% acidity, and 169 mg per 100 gram ascorbic acid. Yield is 94 kg per tree per year. Polyploidy breeding. In this, we will talk about the main aspect, aneuploidy. Aneuploidy have been identified cytomorphologically in progenies from triploid and diploid-triploid crosses. In the higher aneuploids, the plant parts are highly exaggerated and the pollen sterility is very high. The change in morphological traits such as reduction in the amount of growth and the size of leaf distinguish aneuploids from the diploids. Producing triploids will be futile since the fruit shape in triploid is highly irregular and mishapen because of differential seed size. At IARI, with a view to evolve a variety with fewer seeds and high yield, Crosses were made between seedless triploid and seeded diploid Allahabad Safeda. Of the 73 one hybrids raised, 26 were diploids, 9 trisomics, 5 double trisomics, and 13 trisomics. Distinctive variations in tree growth habit and leaf and fruit characters were observed. Aneuploids, particularly trisomics, had promising qualities and may prove useful in developing plants with reduced seedness and in providing dwarfing rootstocks. Finally, future thrusts in improvement. To overcome the high speed content, the technique of endosperm culture may be useful for producing the seedless triploid plants. Plant regeneration from callus culture may be useful for selecting plants resistant to abiotic and abiotic citrus. Recovery of plants of haploid origin from anther and pollen culture offer advantage in breeding program. The breeding potential of wild species is yet to be exploited as they can be source of dwarfing rootstock.